Okay, let's get started. We are talking about translation. Translation. Now remember, let's have a small review of what we have already made clear by this time. So we said that molecular biology, this topic has got what we call the central dogma of molecular biology. So, what is the central dogma of molecular biology? Central dogma of molecular biology. So, the central dogma of molecular biology is telling us that one, DNA is going to make a copy of itself. That is the first thing. And then this DNA copy that we get, we can change it into RNA. To be specific, that is mRNA. And then the RNA can be converted into proteins. Okay. So this is what we call the central dogma of molecular biology. But then each stage here has got a name. The conversion of DNA to another DNA molecule, this is known as replication. And the conversion of DNA to mRNA is known as transcription. The conversion of mRNA to proteins is known as translation. So this is the central dogma of molecular biology. And this one you need to make sure that you know. Now remember, we have already discussed replication, transcription. Now we are talking about translation. So what is translation really? It is making proteins, isn't it? As simple as that. The making of proteins. But then before we get to understand translation, you need to know what are the things that make proteins? What is going to be involved in the making of proteins? So in the making of proteins, this procedure is going to involve some different molecules that we have to know. Okay? Number one, remember when we talked about cells, we said that proteins are made in which organelle? Anyone who remembers? In which organelle are proteins made? Be free to speak. You people don't remember. Okay. So, protein synthesis is going to happen in the ribosome. So, we need the ribosome. Now, a ribosome has got two subunits. Small subunit, okay, small subunit, and the large subunit. Okay, it has got two subunits. Just take note of that. Let me remove this. What other thing do we need? We need the RNA. Okay, and then we need amino acids. So this is basically what we are discussing on this topic. These three things. This is what is going to make up the process of translation. Let's start with the ribosome. Okay, we can start with the ribosome. Now you know that a ribosome has got different parts depending with the cell that we are talking about. Okay, it has going to have different parts. So a ribosome is basically an organelle. It is actually a machinery that is going to be involved in the making of proteins. Okay, very, very important. Take note. Each ribosome is going to have two subunits, the small one and the large one. And the subunits are going to be made up of RNA and proteins. Please take note, all right? Very easy. So ribosomes are what? Have two subunits. Okay? And what are the subunits made up of? They are made up of RNA and proteins. Actually, RNA is of three types, specifically RNA, okay, plus protein. This is what you need to know. Don't forget this one, all right? So now let's talk about it. You have got different types of cells, and you have got different types of subunits. We've said we have got two subunits. So we have got small subunits, okay, and we call these as ribosomal subunits. And then we have got large ribosomal subunits. And then we have got the combined. Now remember again, we have got two types of cells. Teresa, what are the two types of cells of our interest? What's happening? Are we guys revising? So we have got eukaryotic cells or eukaryotes and prokaryotes. Please don't forget. Okay. Now, when it comes to eukaryotic cells, an example of a eukaryotic cell that you can give is an animal cell or a plant cell. These ones are complex cells. A prokaryotic cell is basic. An example you can give is a bacteria. Okay. Now, the small subunits for a eukaryotic cell, you start from 40. If you just know this one is 40S, I just add 20. It will give you 60S. To the 60, add 20. It will give you 80S. So when you combine the 40S and the 60S in a eukaryote, you're going to get 80S. Now, this is not a mathematical addition like 40 plus 60, 100. No. Okay. The S is just representing, it is known as the Sedberg unit. It is representing the sedimentation. Now, to get what is in a prokaryotic cell, just remember that a prokaryotic cell is smaller than a eukaryotic cell. So just to these numbers that you can see on top, just be removing a 10. So the small subunit is going to be the 30S. The large subunit is going to be the 50S. And then if you combine 30 and 50, you are going to get 70S. Okay, as easy as this. What are we saying? What does S represent? S is equal to, we are calling this as a Sandberg unit or the Sandberg constant. Let's say Sandberg unit. Okay. And what does the Sandberg unit mean? The measure of the rate at which a given particle sediments. You know, to sediment is when you put something in a test tube and then you start spinning the test tube. How much is that thing going to settle at the bottom? Okay, that's what we call sedimentation. So this one measures the rate at which a particle sediments. Okay, so this is the first thing that I want you to understand. Okay, now that we have talked a bit about the ribosome, let's talk about RNA. Let's talk about RNA. Now, as we know, RNA is being formed from DNA in transcription. But then RNA is of three types. Okay. The first one is known as M, which is messenger. So messenger or mRNA, that is the first one. The second type of RNA is known as transfer or tRNA. The third type is known as R. The R actually represents something. Let me write it. So that is ribosomal. Ribosomal, also known as RNA. Okay. So we already established, remember earlier on, we said that a ribosome is made up of RNA. Here, we said that a ribosome is made up of RNA and protein. So that RNA is this one here. Now you need to know the function of each one of these. So mRNA carries the genetic code. Carries the genetic code. So what is the genetic code? The genetic code is simply a set of instructions that will direct what type of protein you are going to form. And the genetic code is made up of what? It's made up of what we call codons. Okay? Made up of codons. Take note of that. You understand what codons are. What is transfer? What is the function of transfer RNA? This one transfers even from this one is going to transfer amino acids, and you are transferring the amino acid in order to make proteins or for polypeptide synthesis. Yes, Teresa. Okay, I'll repeat. And then the ribosomal RNA is part of the ribosome that makes proteins. Okay, that makes proteins. So you need to know the functions of these types of RNA. So we are saying that messenger mRNA or messenger RNA carries the genetic code. The genetic code is made up of codons. Now, at this point, you might not really understand what a codon is and what we are mentioning as a genetic code. But don't worry, you understand this very, very soon, just soon. Okay, let's talk about the steps. This thing I'm showing you here is what we call the genetic code. They'll bring this in the exam. You need to know how to read it. Okay, so we'll talk about how to read this one. Now, translation has got three steps. Okay, so translation has three steps. The same steps, remember in replication and transcription we discussed steps that all those are the same. The first step is step one, which is initiation. Initiation. How are we spelling initiation? Okay, initiation. The second step is elongation, can also not be known as polymerization. The third step is termination. These are the steps for all the three steps of central dogma of molecular biology. So now let's start. Please, we are not wasting time in In few minutes we should be done with everything. So make sure that you're understanding, all right? And ask if you're not good. So in initiation, remember mRNA was formed where? mRNA was formed back in the nucleus. Okay, in the nucleus. These three types of RNA. Take note that rRNA is formed specifically in the nucleolus. The nucleolus. All right. Now that we have formed RNA, what is going to happen is that the RNA is going to come out of the nucleus. Where does it go? It is going to go into the cytoplasm. All right. Please take note of that. This step is happening where? In the cytoplasm. Actually, everything is happening in the cytoplasm. Let me just write that. Okay, so these are happening in the cytoplasm. Now let's see what happened. This is a cell. This is a cell here. And whether it's a prokaryotic cell or a eukaryotic cell, let's say it's a prokaryotic cell. And you know that for a prokaryotic cell, RNA has been made in the cytoplasm. It's now going to move out of the cytoplasm into the cytosol. Okay, this is the mRNA. Okay, take note of that. Now that mRNA has moved out of the cytoplasm, what is going to happen? Let's go to the first step of translation, and that is initiation. So what is going to be happening is that immediately the RNA comes out of the nucleus. Remember, the RNA has got, let me write it has got two ends. It has got a 3 prime end, and it has also the 5 prime end. And I want you to really understand this. For this one, we are starting from the 5 prime end going to the 3 prime end. Okay, please, 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 please understand this. Okay, so what is going to happen? Once the RNA moves out of the nucleus, it's now in the cytoplasm. This RNA is going to do something. What is going to happen is that it is going to meet the small ribosomal subunits. That is going to be the first thing that happens. Okay? Please take note. Let me say that again. Once the RNA, during initiation, mRNA is going to be found, or it finds whatsoever that happens, because it went in the cytoplasm. So it is going to find the small ribosomal subunits, all right? So when it locates the small ribosomal subunits, with the help of some proteins called the initiation factor, they are going to bind. So you are going to see now they have bound. So let's say you have got now something like this. Okay, so there it is. So what is this guy? This is known as the small ribosomal subunit. Okay, so this is the first thing that happens. This binding is with help of what? Initiation factors. Okay, now that we have located our small ribosomal subunit as found the mRNA, what do you need to know? What is going to be the next step that is going to happen? The RNA, I want you to understand this, dear friends. The it's You find different things in different books, but the small ribosomal subunit will start moving in this direction. Okay, it moves in 5 prime to 3 prime direction. Please take note what is moving. It is the small subunits. Okay, so the small subunit is moving from 5 prime to 3 prime direction. Is this clear? Okay, so this guy is going to continue moving. Now remember, mRNA contains nucleotides, right? It contains different nucleotides. So you reach, when the small ribosomal subunit reaches where it is written A, U, G, like if it finds these three nucleotides, it is going to locate AUG. What do we call AUG? This is known as a start codon. What is a codon? A codon is just a group of three nucleotides. You can see adenine, uracil, guanine. This one you need to know by heart. You need to know this one. They'll ask you, what is the start codon? It is AUG. Now, what is going to happen is that when the start codon is reached, there's going to be a very interesting thing that happens. What is going to happen when the start codon is reached is that it is going to send a signal to tRNA. All right, it's going to send a signal to tRNA. And what is going to happen is that the tRNA molecule is now going to find an amino acid. Now, what amino acid are we going to find? So depending with the now what we call the genetic code, if you come on the genetic code has got three numbers. This is the first number, this is three letters, this is the second letter, this is the third letter. So you read these three letters. So for the start codon, you are going to see the first letter is A, the second letter is U, the third letter is G. Can you see where these three letters are meeting? So you just want to see where these three letters are meeting. So you first need to go to the first letter, and then you go to the second letter, and then you pick the third letter. Can you see where these letters are meeting? It is here. So the amino acid methionine, okay, is going to be gotten. So AUG means what amino acid? Methionine. You can even see here. But for this genetic code, the first number is here. So the first letter is A, second number is here. So the second letter is U. Remember it's AUC. And then the third letter is here. So the third letter should be C. So it's here. So AUC. Okay. So where are we having the A's? This was actually a mistake on this genetic code. The first number is supposed to be this side. So AUC is this one. AUG. Can you see this one here? AUG. Can you see the amino acid? It is methionine. They have even written there start. Methionine is the first amino acid that is brought. The first amino acid. So you need to know that. Okay. And what is the codon for methionine? It is AUG. Now, instruction has been sent to tRNA to bring the amino acid methionine. I'm going to write this. This is a very important concept that I'm about to write here. You get methionine if the cell that we are talking about is eukaryotic. Okay. For eukaryotic cell, the first amino acid that we get is methionine. Now about for prokaryotic cells, it is known as formyl. Formyl, also known as methionine. So this is the first amino acid that is going to be brought in prokaryotes. Okay, so what is happening is that the tRNA, now look at this. We are going to get tRNA. So that is known as tRNA charging. Let's talk about the charging of tRNA. It is a very important concept because that's the next thing that happens. Okay, let me first talk about what is going to happen. So tRNA looks like this. I even showed the structure. tRNA is like this. This is the structure of tRNA. Can you see that this arm is much longer? This is known as the 3' end. This one is shorter. This is the 5' end. Please take note of that. That is tRNA. But let me show you the diagram that I put here. So this is tRNA. So tRNA has got different parts. It has got this end, the 3' end. It has got the 5' end, okay, and then it has got the D arm, which is near the 5' end, the T arm, the anticodon. Take note of this anticodon, okay, and then it has got a variable arm there. So it is also like this. I can as well put it like this so that you can see it is very easy. Like that, like this, like that, like that, okay, like this. Now I want you to take note, this is a very important concept also. The 3' end, here the 3' end, this is the 5' end which is short. Can you see that the 3' end has got a sequence ACC? So in other words, you have got the 3' end, and then you have got A, and then CC, and then you are going to go to the 5' end. This is a very important one that I don't want you to forget. So it is at the 3' end is known as the acceptor arm. That is where the amino acid is going to be able to attach. Okay, the acceptor arm. That is where the amino acid attaches. The amino acid will attach here. So let's go now and explain initiation. So when the first amino acid has been read, what is going to happen is that tRNA is going to get that amino acid, isn't it? Okay. So let me now put this here. And we said this is our small ribosomal subunit there. So what is going to happen is that we have read, remember, AUG. Okay. We have read AUG. Say AUG is here. So when we read the AUG, what is going to happen is that we are going to bring in a tRNA. So that tRNA, remember we said that tRNA has got different parts. So specifically, this part is known as the anticodon. Remember on mRNA, we have got the codon, right? We said that we have got codons. Codons are just a group of nucleotides. So what is going to happen is that these guys are supposed to pair up. So the tRNA that is going to bring the amino acid is going to be having the codon. Remember, A pairs up with what? Who remembers? What does A pair up with? We have forgotten. Okay, so A pairs up with U. U pairs up with A. Okay, and then G pairs up with C. So the tRNA that is going to come is going to have this anticodon. like that. Now remember, for this one, what I've done is that I've swapped it. So it is going to change. The 5 prime end is going to be somewhere. The 5 prime end is now here. Why? Because this is the 3 prime end of the mRNA. And then this side also, we are going to have something like that. And then remember, this side, this is the longer one, this is the 3 prime end. And here we have got the amino acid. What is the name of this amino acid? It is either methionine or formimethionine, depending with what type of cell we are talking about. Please take note, you have got the codon and the anticodon. The codon is found on what? On mRNA. So this one has got a codon, and then this one is the anticodon. The anticodon is what pairs up with the codon. I don't want you to forget that. So immediately we bring the first amino acid, initiation is done. We are done with initiation. What is going to be happening next is going to be elongation. Now, another thing that I want you to take note of is that initiation is happening in a different region. Okay. So the first amino acid that is brought in what we call the P site. So when tRNA brings the first amino acid, what is going to happen is that the large ribosomal subunit is now going to bind. Okay? The large ribosomal subunit is now going to bind, and that is going to be, let's go now to elongation. Yes, Teresa? These are nucleotides: uracil, adenine, cytosine. So follow the class on nucleotides if you have not done it, otherwise you'll get confused. Okay? All right. So this is initiation. Now the next step is going to be elongation. So now in elongation, what is happening? Remember, this is still our mRNA here. Let me try to write it a bit straight. This is our 5 prime end. This is our 3 prime end. And then remember, we add the small ribosomal subunit here. And we have brought the first tRNA, which is carrying the amino acid methionine. And when the first tRNA is brought, the large ribosomal subunits are also now going to is also now going to come and connect here. So what I'm putting in black here is known as the large ribosomal subunit. When you combine the large ribosomal subunit and the small ribosomal subunit, you form a ribosome. Okay? So this is a large subunit, and this one is a small subunit. Now, one thing that you need to know is that when you form a ribosome, you have got 3 regions. You have got the E site, the P site, and the A site. Each one of these regions has got 3 nucleotides. Don't forget 3 nucleotides. So the E site has got 3 nucleotides. The P site has got 3 nucleotides. And then the A site has got 3 nucleotides also. Okay. The first amino acid that is brought by tRNA is going to come in the P site. Now all these guys have got meanings. E is meaning exit, P is peptidyltransferase, A is amino acid tRNA synthesis. That's where, okay, that is amino acid. Okay, so that is what is happening. In the P site, this is where we add A. Oh, the pen is also the same. This is where we add A, U, C, like that. So the first tRNA has come here. Let's say this is tRNA. This is the first amino acid. Okay. And the name of the amino acid is methionine. Can you see that the A site is empty? So what is going to happen is that we are going to read the A site. Okay. We are going to read the A site. Let's say, for example, on the A site, we have got nucleotides. What can I get? We can get any nucleotides. Let's say, for example, you have got U, C, C. So you are going to go on the genetic code. You want to find out what UCC means. Okay. The first letter is U. The second letter is C. The third letter is also C, UCC. Can you see the amino acid? It is serine. So if we read UCC, it means that serine is going to be brought. But then you again need to understand serine. So we are, remember, we're starting from the 5' end, and then we are saying UCC, and then 3' end. So you need to know the tRNA that is going to bring the amino acid serine. So that tRNA at is going to start 3' end, and then you know that U pairs up with A, C pairs up with G, C pairs up with G, and then 5' end. So this is going to be the anticodon of the tRNA that is going to bring the amino acid. But I want you to take note of something here. Can you see that serine can as well be brought by, can you see that serine, the same amino acid, has got different codons? Okay, even here, all these 4 codons are bringing serine. Can you see where they are different? This is UUC, UUU, UUA, UU, actually UC. So the first one is UCU, UCC, UCA, UCG. So they are only differing at the third position. So the third position is known as wobbling. So the third position wobbles, and that is known as the Wobble hypothesis. Even when you have got a different tRNA that has got a different letter on the third position, it can still bring the same amino acid that you want, and that is known as the Wobble hypothesis. It's only on the third letter. All right. So take note of that. So that is an codon of the tRNA that is going to come in. So what is going to happen at this point is that when that tRNA comes in, that tRNA comes in is going to be here. So this is the tRNA. It has brought an amino acid. This is the amino acid. When this happens, we are going to connect the 2 amino acids. Okay. What is the name of the enzyme that to connect the 2 amino acids? It is known as peptidyl transferase. So peptidyl transferase, transferase, is that the spelling? Peptidyl transferase forms peptide bonds. What is to form a peptide bond? Is to connect the 2 amino acids. Okay, connect 2 amino acids. Take note of this enzyme. So it is going to form a bond between these two guys. So what are you going to be able to see? You'll be able to see, let me now write it again down. You are going to now see this structure. See still the 5' end, see still the 3' end. Okay, and then you are going to have, I'll still put here, this is our molecule. This is our small ribosomal subunit. Okay, and then we have got our large ribosomal subunit here, and then we have got the three sites: E site, P site, A site. What is happening in the in the P site? So now we have formed a bond. Look at what is going to happen. The green guy that you can see there is formed a bond. This green guy has remained alone, bored here, and then the blue guy, it's amino acid, but take note because it's interesting here. Okay, so what happens is that the bond is going to form. You add this amino acid, the other amino acid, these two are going to connect like this. But what is going to happen is that this guy will actually be the one that is going to be, okay, it is going on top there. So we have formed a bond, and this is what we are going to have. Can you see that we have got two amino acids now? In which site? In the A site. Please take note. Okay, A site. A site. Growing chain is in the so the first connection is going to be made in the P site. Okay, the first connection is being made in the P site, in the A site actually. But the growing chain, after you have formed a bond, is going to continue in the P site. Okay, so this is what is going to happen. Now, when this happens, notice what is going to happen. The molecules here are going to move three nucleotides. Okay, or yeah, once the P and the A site have been filled, can you see that the P site and the A site have been filled? Peptidyl is going to form a bond, and then when that happens, when the peptide bond is formed, the amino acid is going to touch from the tRNA in the P site. Can you see that the amino acid in the P site has detached? It is now away. And then when that happens, in the next step, the ribosome is going to move three nucleotides in the 5' to 3'. And you know, moving three nucleotides is moving what? One codon. Remember, three nucleotides is equal to one codon. Don't forget that. Okay, don't forget that. So, what are we saying? The complex, the ribosome moves three nucleotides. Three nucleotides. In which direction? In the 5' to 3' direction. Look at what is going to happen. Remember I said that there are three nucleotides here, there are three nucleotides here, there are three nucleotides here. So if we move three nucleotides, means that what was in the P site will go into the E site. What was in the A site will go into the P site. And when that happens, let's go now on the next stage. We write that on the next board. So now what we have formed is this. Now that we have moved the three nucleotides, this is still our small subunit, and then we have got our large subunit also on top. But I want you to notice what is happening. Here is our large ribosomal subunit. Okay. And then here is our E site. Here is our P site. Here is our A site. Remember, we moved 3 nucleotides in front, meaning that what was in the, remember the A site, that's where we add this guy with the 2 bonds. So it is now going to go to the P site, and what is in the P site goes into the E site. So it is going to now be like this. Okay. So here is the amino acid. Here is the amino acid. And then the guy that is going to come in the E site is going to be removed. Okay. So the guy that E means exit. So the tRNA that comes here is going to be thrown out. That's how it moves out of the procedure. Remember, this is the 5 prime end. This is the 3 prime end. Can you notice now that the A site is again empty? So since the A site is empty, we are going to again read the nucleotides that we are going to see. Let's say, for example, the nucleotide that we see here is going to be G, U, G. What is the amino acid that we are going to bring? We go back again on the genetic code GUG. Okay. The first G is this one. We go to U and then to G. Can you see the amino acid that we are bringing is valine? So this is the amino acid that is going to be brought. And so the tRNA that is going to bring that amino acid should be a tRNA that is complementary to the GUG. So that tRNA is now going to come here. And this tRNA, remember, it is coming with an amino acid. And what is going to happen is that peptidyl transferase is again going to form a bond between this guy. We actually form a bond between this one which is down and this one. Please take note of that. The first amino acid that we started with is always going to be the one at the topmost. You are going to see some exam questions where we're going to see this same diagram that I've written here. And then they're going to ask you to identify this amino acid which is at the top. Or they actually show you some codons and then they ask you to identify the amino acids. It is always that the one which is on the top is the one that you started with. So again, this is 5 prime end, this is 3 prime end. And then here is our small ribosomal subunit. And what has happened? Here is our large ribosomal subunit. Again, after we form a bond, we are going to move 3 nucleotides. So it means that after we form a bond, remember when you form a bond, this 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 guy is going to detach and go and attach to the one that is in the A site. So meaning that the long chain is going to be where? In the A site, right? But we are now going to move it. We are going to now move one codon forward. So in the P site, this is now where we are going to have this guy. It has now 3 nucleotides. And this procedure is going to continue. What was in the P site to go into the E site and then is going to be exposed. It has been removed. This procedure is going to continue and we are going to form a lot and a lot and a lot of amino acids just like that. That is the process of elongation. But after this process happens, that's the process of elongation happens. Look at this. To move for tRNA to move from one site to another, you need another enzyme. it needs, what's the name of the enzyme? This is known as translocation moving. So you need the enzyme known as translocase. Okay, translocase is going to be required to be able to move. So once everything here has happened, we are now going to have termination. Is there any problem on initiation and elongation? Any problem? Okay. Okay. So please, I don't want you to forget. Remember the only tRNA that came direct in the A site. Look at this. The only, this is the E site, this is the P site, this is the A site. The only amino acid that comes direct into P site is coded by AUG, which is a start codon. The rest go first into the A site. I don't want you to forget this. The only amino acid which was taken first in the P site is methionine. The rest of the amino acids, we are saying that if the S site is empty, we are going to read it, the tRNA brings an amino acid. Again, we move. If the S site is empty, we continue like that. Never, never forget that. Okay? So now, what is happening in termination? In termination, we are actually going to read some sequence. Okay? So again, this is a 5' end. This is the 3' end. There is our small ribosomal subunit. Here is a large subunit. The only way termination is going to happen is that if we read the P site is having the stop codon. So the A site, I mean, this is EPA, right? So now if we see that in the A site, we have got what we call a start codon, then termination happens, a stop codon, then termination happens. Remember here, you have got your amino, your tRNA, and this tRNA contains your different amino acids. Amino acid there, amino acid there, amino acid, amino acid, amino acid, amino acid, amino acid, amino acid. To be able to move the tRNA in the P site to the E site, we need the translocase. To form the bonds between the amino acids, we need peptidyltransferase. But now, if you read any stop codon in the A site, the stop codons, we have got UAA, we have got UGA, we have got UAG. If you read any one of these three, these are known as stop codons. Please don't forget. Remember, we also had the start codon. These are stop codons. So if you read any one of these, a protein which is called release factor is going to come and bind to the A site. Okay. So this is no longer tRNA. This is known as release factor. So when the release factor comes and binds in the A site, then termination happens. And that is how it goes. Okay. That is very important for you to understand. And how is the release factor going to bind here? It only binds if you read either UAA, UGA, or UAG in the A site. And then what is going to happen is that this bond is going to break off here. And then the tRNA is going to go and undergo some modifications. Now let's talk about one important thing. There is what is known as tRNA charging. Okay, tRNA has to be charged. How is the amino acid being added to tRNA? That is what we are calling as tRNA charging or amino acid activation. Okay, tRNA charging or amino acid activation. So tRNA charging is basically getting to get an amino acid and add it to this guy. Amino acid activation will be a bit of similar. So before translation takes place, each tRNA is charged with a specific amino acid. The enzyme that does that is known as amino acid tRNA synthetase. That is amino acid that will be able to add that is the enzyme that has an amino acid to tRNA. What's the name of the enzyme? Amino acid tRNA synthetase. Not synthase, synthetase. There's a difference. Synthetase, it means that it uses energy. Synthase doesn't use energy. So this is the enzyme. So let's say this is the enzyme here. It is, say, like this. Okay. Let's say this is the enzyme. So what is going to be happening is that this guy is going to get an amino acid, but then before you get that amino acid, you need energy, isn't it? You need the ATP. So this guy has got two sites. It has got the site where tRNA is going to be able to bind, and then the site where the amino acid will be able to bind. So let's say, for example, the amino acid binds here. This is the amino acid. Where is tRNA going to bind? tRNA has got also another site on this same enzyme where it's going to bind. Let's say here is tRNA. So what is going to happen is that now that these two guys have attached on the enzyme amino acid tRNA synthetase, then we are going to connect them. So you see, if we connect them, then that means that we have now attached the amino acid to the tRNA and then they can move out. So in that case, we have done what? We have activated the tRNA. This is known as tRNA activation. But then what is happening is that the amino acid also, before we can even attach it to tRNA, we have to attach it to ATP. Okay, this is, I want you to differentiate this because that's how you find questions. This is tRNA charging. Now, what is amino acid activation? Amino acid activation is that you get an amino acid, you put it on this same enzyme, the amino acid tRNA synthetase. When you put it on this enzyme, you now attach ATP to the amino acid. And when you are going to connecting, so now you have got an amino acid, say this is the amino acid. It is attached to ATP. When you are going to be connecting this amino acid to the enzyme amino acid tRNA synthetase, the bond between the ATP will break. So two phosphates are going to go. It is going to remain as one phosphate. It becomes ADP. Now remember, this guy is in the enzyme amino acid tRNA synthetase. And then what is going to happen is that when a tRNA now comes and attaches to the amino acid tRNA synthetase, and then this bond is formed, this now is going to break off, the ADP will break off. Okay, so amino acid activation is basically getting to add energy to the amino acid, and then you are trying to connect it to tRNA. tRNA charging is the actual attachment of the amino acid to tRNA. Does that make sense? Are we making sense? Okay. So just one more thing as we conclude. After the after termination happens, you have formed your compound which has got polypeptide bonds. Now this one has to be activated. I mean, after transcript, after replication, we did have some modifications. After transcription, we had some modifications. After translation, we also have going to have modifications. So those are known as the post-translational, post-translational modifications. Because the polypeptide chain that we have formed do not be able to work. There are some things that you have to add in order for it to function. So what are some things that you do in post translation? Remember, this is translation. That's why I was saying translation. The first thing that you do is phosphorylation. So in phosphorylation, what happens is that the chain that you formed, you're actually going to be adding some form of amino acids which contains phosphate. That is phosphorylation. Okay. And then you are going to have the other thing that you do is methylation. Methylation is basically the addition of a methyl group. So you're going to be adding methyl groups to your polypeptide that you have formed so that it becomes functional. The other thing that you can be doing is glycosylation. Glycosylation is the addition of, you are trying to add glucose molecules or sugar compounds to your molecule. That is glycosylation. And then the other process is going to be proteolysis. So there are actually some form of, when you form this protein, there are some parts which are inactive. Certain proteins are made inactive, and the inactive proteins, we call them zymogens. So those ones, in order to activate them, you are going to use the enzymes, the proteases that are going to cut some bonds to remove what is inactive. Okay, like that, you are going to activate your compound. The other thing that you can do is known as any acetylation. So acetylation is basically the transfer of acetyl groups, o- and it's known as any acetylation because you are adding acetyl groups on the nitrogen end of this chain that you are forming. Remember, the amino acids have got the amino end and the carboxylic acid end, or the carboxyl end. And then the last thing that you can do is lipidation. In lipidation, what you are basically doing is that liquid lipid components are also added. So these are some modifications that you are going to see, and then the protein that you form is fully functional. And this process, my friends, is what we call translation. We are going to do exam questions in the next class when we also talk about the genetic code.